In this question, you have to identify the two functional groups characteristic of sugars. Now, sugars are characterized by the presence of functional groups such as carbonyl, which is found as either as aldehyde group in aldoses or ketone group in ketoses. For example, glucose is an aldose, but fructose is a ketose. And another functional group, that is the hydroxyl group. Now, sugars are polyhydroxy compounds, meaning they have multiple hydroxyl groups. And these two groups define the structure and reactivity of sugars, influencing their properties like solubility and chemical reactivity. So the correct answer is option C, carbonyl and hydroxyl. In this question, you have to identify what type of substance is concanavalin A is. Option A, an alkaloid. Alkaloids are nitrogen containing organic compounds which are usually found in plants, but concanavalin A is not an alkaloid. Hence, it is incorrect. Option B, an essential oil. Now, essential oils are volatile aromatic compounds which are also extracted from plants, and they are often used for fragrance and flavor. Whereas concanavalin A is not volatile and it also lacks the aromatic properties. Hence, it is incorrect. Option C, a lectin. Now, concanavalin A is indeed a lectin, and lectin is a type of protein that binds specifically to certain carbohydrates, and it is widely used in biochemical studies to bind the carbohydrates. Thus, it is the correct option. Option D, a pigment. It is also incorrect. Now, pigments are substances that impart color, but concanavalin A does not have any color imparting properties. Hence, it is incorrect. In this question, two statements are given, and based on these two statements, you have to choose the correct option. Now, statement A, coenzyme or metal ion that is tightly bound to enzyme protein is called prosthetic group. It is true, because a coenzyme or metal ion that is tightly bound to an enzyme is indeed referred to as the prosthetic group. Statement B, a complete catalytic active enzyme with its bound prosthetic group is called apoenzyme. Now, it is false, because a complete catalytically active enzyme with its bound prosthetic group is called a holoenzyme, not an apoenzyme. An apoenzyme refers to the inactive protein portion of the enzyme without its prosthetic group or cofactor. Hence, the correct answer is option B. In this question, you have to identify the purine molecules which are found both in DNA and RNA. Option A, adenine and thymine. Oh, it is incorrect. Adenine is a purine, but thymine is a pyrimidine. And thymine is only found in DNA and not in RNA. Option B, adenine and guanine. Now, adenine and guanine are both purines, which are nitrogenous bases with a heterocyclic double ring structure, and they are present in both DNA and RNA. Hence, it is the correct option. Option C, guanine and cytosine. It is incorrect, because guanine is a purine, but cytosine is a pyrimidine. Option D, cytosine and thymine. It is also incorrect, because both cytosine and thymine are pyrimidines, and thymine is found only in DNA and not in RNA. In this question, you have to identify the basic amino acid. Option A, lysine. Now, lysine is a basic amino acid because it contains an amino group in its side chain that readily accept protons, giving it a basic character. And it has positive charge at physiological pH. Hence, it is the correct option. Option B, valine. This is incorrect because valine, which is a non-polar hydrophobic amino acid, does not have a basic or acidic side chain. So it's a neutral amino acid. Option C, tyrosine. This is also incorrect. Tyrosine is a polar amino acid with an aromatic side chain, but it is not basic. It has hydroxyl group in its side chain and can be weakly acidic. Option D, glutamic acid. It is also incorrect because glutamic acid is an acidic amino acid due to the presence of carboxyl group in the side chain, which can donate protons, giving it an acidic character at physiological pH. In this question, you have to identify that Floridian starch has structural similarities to which compounds. Now, Floridian starch is stored food material in the red algae and it is highly branched and resembles amylopectin, which is a component of the starch, and glycogen, which is also known as animal starch. And all of these are branched polysaccharides. Hence, the correct answer is option D. In this question, you have to match the components given under column 1 with that of column 2. Now, malonate is the competitive inhibitor of the catalytic activity of succinic dehydrogenase. Thus, A matches with 2. A collagen, which is proteinaceous in nature, possesses peptide bonds. Thus, B matches with 4. Now, chitin is a homopolymer that is present in the cell wall of fungi and exoskeleton of arthropods. Thus, C matches with 3. Arbine and ricin are toxins which are secondary metabolites. Thus, D matches with 1. Hence, the correct answer is option C. In this question, you have to identify the substances having glycosidic bond and peptide bond respectively in their structure. Option A, cellulose and lecithin. Now, cellulose is a polysaccharide which is made up of glucose units which are linked by glycosidic bonds. But lecithin is a phospholipid, so it doesn't have any peptide bond. Rather, lecithin has ester bonds that links the fatty acid to the glycerol moiety. Hence, it is incorrect. Option B, inulin and insulin. Now, inulin is a polysaccharide which is made up of repeating units of fructose which are linked by glycosidic bonds. And glycosidic bonds are specific type of covalent bonds that connect the monosaccharide units in a polysaccharide. And insulin is a protein, and proteins are composed of amino acid which are linked together by peptide bonds that forms between the carboxyl group of one amino acid and the amino group of another. Hence, this is the correct option. Option C, chitin and cholesterol. The chitin is a polysaccharide like cellulose and it is composed of N-acetyl glucosamine units linked by glycosidic bonds. But cholesterol is a lipid, specifically a steroid, and it does not have glycosidic bond or peptide bonds since it is neither a carbohydrate nor a protein. Hence, incorrect option. Option D, glycerol and trypsin. Now, glycerol, which is a backbone of triglycerides and phospholipids, but it doesn't have glycosidic bonds. And it is involved in ester bonds when it forms the parts of fat. So it doesn't fit with the glycosidic bond or the peptide bond that is in question. And trypsin is a protein which is a digestive enzyme. And like all proteins, it contains peptide bonds between amino acids. Hence, it is incorrect option. In this question, you have to identify the most abundant protein in animals. The option A, lectins. Lectins are carbohydrate binding proteins found in a variety of plants and animals, but they are not most abundant protein in animals. Hence, it is incorrect. Option B, insulin. It is also incorrect. Insulin is a hormone which is involved in regulating blood sugar levels, and it is not the most abundant protein in animals. Option C, hemoglobin. Now, this is also incorrect. Though hemoglobin is an essential protein in RBCs, which is responsible for transporting oxygen throughout the body, but it is not the most abundant protein in animals. Option D, collagen. Now, collagen is the most abundant protein in animals, which makes up about 25 to 35% of the whole body weight of the protein content. And it is a structural protein which is found in connective tissues like skins, tendons, ligaments, cartilage, bones, and it provides strength as well as structural integrity. Hence, it is the correct option. Secondary metabolites such as nicotine, strychnine, and caffeine are produced by plants for their option A, defense action. Now, secondary metabolites like nicotine, strychnine, and caffeine are primarily produced by plants definitely as a means against herbivores and pathogens. That is defense action. And these compounds can be toxic, repellent, or deterrent to animals and insects, thus helping them to protect from being eaten or being infected. Hence, it is the correct option. Option B, effect on reproduction. Now, while some secondary metabolites can influence reproductive processes, such as attracting pollinators or even deterring herbivores, that may harm the reproductive structures, but the primary purpose of these compounds is not related to reproduction. Hence, it is incorrect. Option C, nutritive value. Now, secondary metabolites are generally not produced for their nutritive value. In fact, they may be harmful or toxic to herbivores and are not intended to provide nutrition. Hence, it is incorrect. Option D, growth response. Secondary metabolites also do not regulate growth responses in plants. Hence, it is also incorrect.